I'm Dr. Jean Stutz, and I am a faculty member in the Science and Mathematics section of the College of Letters and Sciences. And I am a professor here at ASU, and I work with mycorrhizae. So I'm really excited today to talk to the structure and function class about mycorrhizae. So when we start thinking about mycorrhizae, it's really seeing roots in a different way. When we learn in introductory bio about roots, we think about roots just totally being plants. But in reality, they're really mycorrhizae. And mycorrhizae are a symbiotic relationship between plants and roots, and they occur in approximately about 95% of all land plants. There are some plants that are non-mycorrhizal. For example, plants in the brassica, like Brussels sprouts, cauliflower, those, and some weeds of brassica are non-mycorrhizal, but most of the rest of the plants do have this relationship. So when we look at this relationship, this diagram on the bottom here shows that it's a relationship between the plant and fungi, but it's also a relationship with the soil. So the fungi are both inside the roots and outside in the soil, and that helps with the symbiotic relationship. So mycorrhizae are an example of the symbiotic relationship that's called mutualism, and mutualism benefits both partners in the relationship. So the benefit to the fungus is that the fungi absorb carbons from the plant. So all of their energy comes from the plant. And some people who work with mycorrhizae have a mycorrhizal fungal perspective, and they have, one of my friends has a quote that says, "Plants are just photosynthetic appendages of fungi." So if you're thinking about it from the fungus point of view, the plants are what provides the carbon to them. Now from the plant point of view, the mycorrhizae increase the growth of the plant. Every time people give a lecture or presentation about mycorrhizae who work with them, they always have a big plant, little plant slide, and this is mine. So here we see pine trees, and we have inoculated plants on the right, and uninoculated plants on the right, and inoculated plants on the left. And you can see that the plants on the left that are colonized by the fungus underground are bigger than the non-mycorrhizal plants. And this is because the fungus helps the plant take up nutrients. It also increases their stress tolerance, which is especially important here in the desert, and I've done some research on this, that it affects the drought tolerance of the plants, increases their drought tolerances. It can also increase their resistance to plant pathogens. And importantly, in the desert, it can increase their survivorship, how they survive under adverse circumstances. So I want to go a little bit more into the relationship underground and talk a little bit more about how mycorrhizae help in nutrient uptake. So there's fungi, their vegetative body is called mycelium, and they produce filaments called hyphae. And these hyphae ramify through the soil. As we can see in the diagram on the left, they're coming out of the root and going into the soil. And they grow in between the soil particles, in the spaces between the soil particles. And they increase the surface area of the root. So if we just would look at root hairs, which are also involved in nutrient uptake, they only can go for a small distance from the root. But the mycorrhizal fungi can go much further out into the soil and pick up nutrients. And this is especially important for picking up the nutrient phosphorus, because phosphates that are in the soil do not move around very much. So they have to come in correct contact. So you can see on the diagram in the right, they're hyphae picking up phosphates and then transporting them to the root. And then there's a relationship inside the cell for this particular kind of fungus, and they're released into the root and then taken into the rest of the plant as a nutrient. Now you can see also that the plant is providing sugars to the mycorrhizae that's out in the soil that allows it to grow. There are some mycorrhizae that also use enzymes to break down macromolecules, so like organic matter in the soil. And this helps in taking up nitrogen because they can absorb, if they're breaking down proteins, they can absorb the amino acids and then bring those into the host plant and supply nitrogen to the plant. Another really important thing about mycorrhizae is they form what's known as a common mycelial network between plants. So we can see here that this network can be between plants of the same species, which is on the left. We can see some mycelial metric of an established pine, and we can see some germinating pine seedlings growing into that network. So this really helps new seedlings, when they tap into that network, they start to share carbon with the other more adult plants. And we actually think that this is really important as a possibility in deserts for plants that, I know you've learned about plants that are nurse plants for cacti, and they provide things like shade, but we also believe, based on some research that was done down in Baja California, that those nurse plants transfer carbon to those little tiny cacti as they start to grow. And this helps them in their growth as they become bigger. And then eventually they start producing most of the sugars they produce themselves. But if you recall, cacti are very, very little when they first germinate. We can also have a common mycelial network occurring between plants of two species. So on the right, we can see that we have a clover plant and a corn plant, and they can share nutrients too. Now, there are two main types of mycorrhizae, and they're called Endomycorrhizae and ectomycorrhizae. And if you recall, endo means inside and ecto means outside. So this has to do with not whether they're inside and outside the root, but whether they're inside and outside the plant cells in the root. So the Endomycorrhizae actually are inside cortex cells. So the diagram we saw a couple minutes ago, that was an example of an Endomycorrhizae because we had a structure inside of the cortex roots. But we also have hyphae and spores that occur outside the roots. And the Endomycorrhizae are the most common and ancient type of mycorrhizae, and most plants form this type of mycorrhizae. And the interesting thing about it is the roots, unless they are stained with a stain to see the fungus, they don't look too much different externally than roots that don't have mycorrhizae. So it's sometimes difficult to tell without staining if the plants have this association. In contrast, the ectomycorrhizae occur mostly in plants that are conifers. So we see the pine tree here has an ectomycorrhizae, and the roots actually that are colonized by the fungus look different. We have a change in the morphology, and we actually have a structure on the outside of the root, but we also have material inside the root. Okay, so I want to talk a little bit more about these two types, the first type I want to talk about is an Endomycorrhizae, and it's called Arbuscular Mycorrhizae. And this group of fungi actually are very ancient, and they are believed to have influenced the evolution of plants as they came onto land. And they actually have their own group of fungi called the Glomeromycetes. So the Glomeromycetes contain all of these Arbuscular Mycorrhizae, and their name comes from the fact that they form structures inside of the root cortex. So we can see in the middle diagram when we stain roots with a stain called trypan blue that binds to the cell wall of fungi but not plants, that the structures become visible. And the arbusculars can be seen as these branch structures inside the cortex root. Now these root cells are living, so we have exchange between that living root cell and the living fungus cell right inside of the cell wall of that cell. And we have sugars going out and nutrients coming in. There are over 200 described species of this particular group of fungi, and it's based on the identification of spores. Most of the identification of fungi are based on their spores. So we can see some of the spores forming on the outside of the root in the diagram on the top. Now some of these species form vesicles inside the root, and we can see them on the bottom right. And those vesicles are storage structures. They store lipids for the fungus. And the fungi that form them sometimes are called vesicular arbuscular fungi, or VAM. And this is kind of an old name for Arbuscular Mycorrhizae. but you might see that if you start looking up these fungi. So as I said before, this is the most common type of association, and over 80% of the land plants, including a lot of herbaceous plants and woody plants, form this association. So it's very common in areas like grasslands and deserts, and it's also the main form of association with agricultural plants. Now, moving on to the Ectomycorrhizae. These are formed mostly by another group of fungi called the Basidiomycetes. And the Basidiomycetes, if you recall from Introduction to Bio, are the fungi that include the mushrooms. And very often these fungi form mushrooms above ground certain times of year, and a lot of these are edible mushrooms. So people who are mushroom hunters know to go back to certain trees year after year because those edible mushrooms have an association with that particular tree. They alter the root morphology. So we can see in the center image, this is some mycorrhizae that are associated with cottonwood in riparian areas. One of my students took this image. And you can see that the roots are kind of inflated and fatter where we have the association. And this is because the fungus forms a mantle, what's called a mantle, around the root. So in the bottom right-hand side, we can see a cross-section of a root with that mantle. So that mantle protects the root from pathogens, and also is involved in being associated with the fungus outside of the root, which is picking up the nutrients. Now, in contrast to the Endomycorrhizae, the exchange occurs in what's called the heart-tick net, which is a structure that occurs between the cortex cells in the root. And this is where we have the exchange. So these fungi do not go inside of the root cells themselves. And the hosts in this case are mostly gymnosperms. So we see this a lot in boreal forests where conifers predominate, and also can be other woody angiosperms. And we've studied this association in riparian areas in Arizona, and it can include, the host can include cottonwood and willow. Now there are some other types that I want to mention, some other types of Endomycorrhizae. There's an association with orchids and with fungi and the orchids that are very involved in helping the seeds of orchids germinate and the little plants grow, the little seedlings grow. There are also some other types of mycorrhizae called albuteroid and ericoid mycorrhizae, which form in areas that are very cold and wet. And this includes the heather and heaths. And the fungi involved in this case are Basidiomycetes and Ascomycetes, and then we have plants in the family Ericaceae that are involved. So these are very minor types of mycorrhizae, probably involving less than 1% of the plant species. And finally, there's another group of fungi that was recently, in the last 40 years, discovered to be associated with plant roots. And these are called dark septate endophytes, or DSE. And these fungi are mostly in the Ascomycetes. So you might recall that the Ascomycetes are sometimes called the sac fungi, and they are fungi that form ascospores. And for many, many years, people have been seeing these dark structures, mostly on the outside of roots, with septa in them, and septa are the cell walls. So you can look at the right-hand side. Here's an example of what the dark septate structures look like, and these form on and inside the roots. And what we didn't realize is that inside roots, we also have more hyaline or non-colored hyphae, and that doesn't stain with the standard stains that we use for mycorrhizae. So a new staining technique was developed in the early part of this century that we could visualize these fungi, and my lab was one of the first groups to look at these in desert and arid ecosystems, and we found that a lot of the shrubs and grasses in the arid ecosystems are colonized with these particular fungi. And it appears to be especially prevalent in plants in stressful environments. We really don't understand totally how, because this is a relatively new discovery, how these fungi contribute to things like acquisition of nutrients and influence plant growth. But we do believe that they might be effective in helping the plants with water stress and also in tolerating root pathogens. So this is an initial study that showed that looking at a grass, a panicum grass, and it was looking at if you inoculated that grass with a root pathogen called fusarium, and you had non-DSC roots, you had lower growth and more disease. But if you look on the right, you see a combination of the fungus, the DSC fungus and fusarium, and you can see that the plants are growing better and there's less root infection. So we do know that they have a role in in suppressing root pathogens and in tolerating stress. So what kinds of mycorrhizae occur in the arid southwest? What are adapted to our area? In the deserts, we are seeing the AM fungi, the arbuscular fungi, and the DSE fungi. But as we move up into our more forested areas, we start to see the EM fungi being prevalent. So the diagram on the left is showing why this happens. As we increase in latitude or altitude, and in Arizona we're increasing in altitude, we get different types of plants that produce different kinds of nitrogen in the soil. So we go from more mineral soils with less organic matter, and nitrate being the predominant form of nitrogen, which we would see in grasslands and in the deserts, to more organic soils, which we'd see up in the forest because we have a lot of leaf litter that makes organic matter in the soil. So in those types of situations in the middle here, where we start to go up to our ponderosa pine forests, we start to see EM fungi. As we go up in latitude mostly and go into colder regions, we get into and wetter regions, we see those heathlands where the ericoid mycorrhizae can predominate. So if we look at the map, a worldwide map of the distribution of the two main types of mycorrhizae, the ectos and the AMs, we can see that in areas where we have deserts and grasslands, it's mostly AM fungi, and where we have forested areas, it's mostly the ectomycorrhizae predominate. So why do people study mycorrhizae? They study mycorrhizae because we are increasingly realizing that mycorrhizae are important in plant diversity and functioning in natural ecosystems. And we also study mycorrhizae because they do have an impact on agricultural plants, and as we try to find more sustainable methods of growing plants with less fertilizer, these mycorrhizae are increasingly becoming important, studying them. And we also study them because they have a role in the restoration of degraded ecosystems. So if an ecosystem is degraded, if there's no plants growing there, or if the soil has been disturbed, we are disturbing the mycorrhizal symbiont, the fungi, and sometimes we need to re-add those fungi back into the soil to get that restoration to occur. So I've actually studied mycorrhizae in all of these settings, and I'm going to talk a little bit about number 1 and number 3 as we finish up this talk. So I'm going to talk about mycorrhizae in riparian ecosystems. So riparian ecosystems are the interface between aquatic and terrestrial environments. And in Arizona, these are very important as hotspots of biodiversity. Even though they are an estimated about 5% of our land area, they are probably about 50% of the diversity of bird species and plant species and everything occur in those areas. So I've had some of my students in the last 15 years or so working in these riparian systems. Actually, it's 20 years now, but they've been working in these riparian systems, and the circles show some of the areas that we've worked on. We've worked on the San Pedro River, up in the Gila River, up on the Verde River, and also on the Salt River. And we're really interested in what the impacts of humans are on riparian ecosystems and the mycorrhizae in them. So humans have really altered these riparian ecosystems by diverting water for agricultural and uses in cities, and also by damming up areas. So we've reduced the amount of water in riparian ecosystems throughout our state. Now, if you look at the vegetation across a riparian area, you can see that there's different vegetation as we go across the space from what's called the channel, or where the open water is going through. So if we go from the right and go to the left, we can see right next to the water we have a lot of cottonwoods and willows that live. We also have some grasses. There's some that we've been studying called Sacchaton grasses. Above, this is usually called the first terrace or the lower terrace. And then usually there's what's called an upper terrace. Very often this is an area where we have what are known as mesquite bosques. We've studied those areas. And then finally, as we get out from the river, we get Sacchaton grasslands and natural ecosystems. Now, a lot of the upper terrace Sacchaton grasslands, as people started to colonize, for instance, the San Pedro River and also the Salt River, these are flat areas, so they became prime agricultural areas. So most of those areas within the 20th century were farmed. And especially down in the San Pedro River, a lot of those areas have been abandoned as farmland for a variety of reasons. And now we're working on trying to restore those areas. So first I want to talk about what we found about about the riparian plants and what we found. We found that some of the riparian plants can actually associate, and this was in the literature beforehand, but we found in the East Coast, but we've also found this in our western rivers. They can be associated with both ecto and AM fungi. So this is showing some images from cottonwood and willow, looking at the Endomycorrhizae on the right and the Endomycorrhizae on the left. And the amount of colonization appears to be dependent on the age of the plant, the season of the year, and water availability. So as the plant ages, it usually starts out, these cottonwood and willow plants usually start out along the banks of the river, in bars that are deposited during floods. And the very young plants, because that is very mineral soil, are predominantly Arbuscular Mycorrhizae. But as they age and we start to accumulate litter and organic matter in the soil, they switch over to Ectomycorrhizae. We're also finding that water availability can really affect the amount of mycorrhizae in the roots. So this is a study by one of my graduate students, Vanessa Beecham. She was looking at different reaches of the Verde River, and we were looking at reaches where there were dams, which are called regulated reaches, and unregulated reaches, which are areas which are not dammed, where we have free flow. And we had paired areas along the Verde from up near Paulden all the way down to the Salt River. And one of the things we found is in all of those paired reaches, in the unregulated reaches, we had more mycorrhizae than in the regulated reaches. So this is of real concern. As we start to dam, we might be decreasing the colonization of these essential fungi and therefore decreasing the growth of the plants. We also wanted to look at the communities of the mycorrhizae and how that might be influenced by how far we get from the channel. And we found that there were actually two groups of fungi. So this is on the left, we're showing a little bit about the classification of these fungi. And most of the fungi that we see in arid areas are in what's known as the order Glaminea, and that includes the family Glamaceae and the family Aclospiraceae. And these fungi are really, especially the fungi in the Glommeraceae, are kind of small white fungi that are found primarily in the Sonoran Desert, and that's the prevalent thing that we see in the desert. Now, as we get closer to the channel, we start to see the occurrence of these other group of fungi, and we actually found some really interesting new species. So if we look at these fungi on the graph from one of Vanessa's papers, we can see that it is showing in the plots that are closest to the river, which are black, that we have certain fungi that occur more often in the black is actually, the black is actually furthest from the river. The white is closest to the river. So we can see in the white, where we have white occurring, that we have this group of fungi, mostly in the Aclospora on the left, that occur closest to the channel. And as we go to the right, we see more and more fungi that are occurring further from the river. So another type of research we've done in riparian areas is looking at these Sacchaton grasslands. As I said before, these are common in upper terraces of riparian areas, and we can see an image on the right of what a natural Sacchaton grassland looks like. This is, and the big plants in the background are actually cottonwoods on the channel. So this is a wide expanse of area up from a riparian area that traditionally was in grasslands. Matter of fact, when the Spanish explorers came through southern Arizona, they talk about these giant grasses that came up to the shoulders of their horses as they were going through the area. As I said, most of those were converted into agricultural areas because they were close to water and flat. But as these become abandoned, there's been a lot of work on trying to restore these grasslands. And there's been some difficulty in doing so. So I had a few graduate students that were involved in, first of all, looking at whether there were mycorrhizae in this area, because a lot of the plants that were growing in the area after the fields were abandoned were these weedy species that were non-mycorrhizal, like tumbleweed and mustard. So we wanted to see how much mycorrhizae was there, and if you inoculated Sacchaton plants with the mycorrhizal fungi, if they would grow better. So here's some of our results on that. So we found that as far as looking at the inoculum, or the amount of inoculum in abandoned agricultural fields, we found it was very similar to grasslands, but the communities were altered. So it seems like we had maybe more beneficial communities in the grasslands than we did in the abandoned agricultural fields. And then we actually inoculated Sacchaton transplants and put them into abandoned fields as part of a restoration project down in Patagonia. And this is some data from a published paper from a student of mine, Brantley Richter, and we found that the growth was definitely increased in the plants that were inoculated versus the uninoculated plants. And what we're hoping is, as part of a restoration effort, to have little islands of mycorrhizal plants and having that association spread through those restored areas as the common mycelial network. Okay, in summary, I just want to summarize what I've talked about. We are now realizing that most plants' roots are colonized by mycorrhizal fungi, and this symbiotic relationship helps plants take up nutrients and water, resulting in increases in growth. The common mycorrhizal fungi in the Southwest include the arbuscular, the ectomycorrhizal fungi, and the dark septate endophytes. And human activities such as water withdrawals and agriculture we're increasingly find can affect and impact negatively, in some cases, the mycorrhizal communities and might affect plant growth. So I'm happy to speak to you today about this topic.